All right, hello everyone. Looks like we're having some people join. I'm Maggie Flanagan, Assistant Professor of Pathology and Neuropath Core Leader at Northwestern's Alzheimer's Disease Research Center. And today we are having our second HALO webinar session, which is specifically focusing on HALO modules applied to neuropathology research. So just to start with a disclaimer, this event is not sponsored by HALO, nor is NACC endorsing HALO through hosting and publishing this webinar. The goal of this webinar is for members of the Digital Pathology Working Group and our speakers to share their personal experiences with using the HALO software and to educate others on the software. So I'll just briefly introduce speakers and panelists for today. So we have Dr. Dirk Keene, Associate Professor of Laboratory Medicine and Pathology, and Nancy and Buster Alvord, Endowed Chair and Director of UW Neuropathology, and the Associate Director and Precision Neuropathology Core Leader of the UW ADRC. We also have Jeanelle Azira Torres from the Keene Laboratory, who's a research scientist at the University of Washington. Dr. Russ Huber, Assistant Professor of Neurology at Boston University and Director of the PTSD Brain Bank, and Alyssa Myers, Application Scientist from HALO at Indica Labs. So we will start off with Jeanelle presenting on HALO modules applied to neuropathology research. She will provide an overview on some of the most commonly used HALO modules and add-ons for quantitative tissue analyses in neuropathology specific research settings. These include Multiplex IHC, in addition to machine and deep learning classifier add-ons. Following her presentation, Alyssa Myers, the application scientist from HALO, will give a brief update on the recently released HALO 3.3 software update, and this will include new features as well as a summary of some of the bug fixes. And lastly, we will wrap up the session with a live question and answer session with all panelists. So you can feel free to submit your questions throughout the presentation and we will address them at the end. So take it away, Jeanelle.
Thank you. Good morning, everyone. I'm going to share my screen. And okay, so as Margaret introduced, I work for the Brain Lab at the University of Washington. And today I'm going to talk about the HALO modules and how we apply those modules for the neuropathology image analysis at the University of Washington. Last time I spoke about 4 products. Today I'm going to talk about the HALO AI with their machine and deep learning techniques and approaches, and how we can combine those techniques with the HALO modules. So to just refresh the knowledge of everybody, and HALO AI refers to the use of artificial intelligence. Artificial intelligence is the branch of computer science that is in charge of dealing with the stimulation of intelligent behavior in computers and the capability of machine to let the machine imitate a human like behavior. There are 2 sub-branches or subfields inside the artificial intelligence. One is the machine learning, which is a subfield of the AI, and is the concept that a computer program can learn and adapt to new data without the human interference. And they can be, you can train those using supervised learning and unsupervised learning. The other subfield that I'm going to refer the most is the deep learning approach, and this deep learning is also called a convolutional neural networks, and it's a subset of the machine learning. And they it takes the inputs image assign importance, so it's really important for the deep learning approach to use a hierarchical way training, and the important important thing is that the images images are easy to process and without losing the features which are critical for getting a good image prediction or classification prediction. So in HALO, they refer, there are two aspects. They call it the tissue classifier, which uses the random forest technique in machine learning, and the deep learning that uses, as I mentioned before, the neural networks, and they create these several masks that I'm going to explain briefly during my presentation. So there are two different ways that work. Machine learning uses the random forest. So in this one, that creates a decision trees from random selected subset of trainings. And in this one, the input layer enters to the process of several hidden layers to do an output layer to do the classification of the tissue. So the deep learning, as I mentioned, they use a hierarchical way to classify the tissue. So it's very important that you take the time to determine what are the ranks of your different type of annotations or regions of interest. Then the computer divide those annotations that you use for training, and they divided into small patches of pixels. And once they is trained to recognize, you can annotate run these annotations, pre-trained annotations, to unseen whole slide images to get your desired analysis. So the modules that I'm going to talk about today are the Multiplex IHC, which is quantify the expression up to 5 immunohistochemistry markers into the nuclei, cytoplasm, and membrane. They can work also in combination with the color deconvolution module and the AI to define the region of interest and pre-trained nuclei masks. The type of data that you can obtain are the area quantification of your region of interest, and also the area, the data, cell-by-cell data. For example, we use it for differentiate inclusions or positive stain in the cytoplasm nuclei, or the ones from the ones present in the neurites. We use it mostly for doing the analysis or for the 88, for the TDP43 or the NUN. Other module that we use is object colocalization, and this can measure one or two objects staying with different chromogens. It can count objects and stain intensity for each object and quantify the object colocalization when it's applicable. It gives you the total and single object information, for example, diameter, area occupied by the staining, and it's useful for heterogeneous shapes of stain like GFAP, 6010, IBA1, TDP43, and CD68. As an example. And the last module is the microglia activation. It measures, as the number says, the activation of the microglia cells. Detects the microglia soma and processes, gives the report of the number in activated and activated microglia cells and the number of negative and not, which are non-microglia cells detected. The reports process also gives you the information of the processes area, the length, and the processes branching, and is specialized in microglia stain like IBA1, CD68. So I'm gonna show you how that work. So last time I spoke about how to use HALO to do the manual annotations. This time I'm gonna show you how to do these annotations using the classifiers modules, the classifiers masks or networks. I mentioned that one of the ways to approach to this is using the machine learning technique called random forest. Random forest is very, in this case, in HALO applications, they use is very useful because it's easy to train. however, you can use it only for stains or regions of interest that are very distinct one from another. For example, the color, it uses features like the color or the texture of the tissue. So I can separate the regions of interest, for example, in this case, between gray matter and white matter. So I'm gonna, for the sake of this webinar, I'm gonna show you the pre-trained model that I did. So in this one, for example, we can annotate different aspects of the my region of interest. So there are two aspects that are really important in all the classifiers. One parameter that is one of the most important ones is the resolution, which gives you the information to the computer on how tuned or how fine he can go with the recognizing those areas of interest. So in the resolution, you can go as high as possible where the object of interest is still discernible with your eyes. So that's one key. The other aspect is the minimum object size in microns. So in this one, this parameter merges areas of pixels in a class smaller than this value, so into the dominant neighboring class. So for example, if I am going to analyze this big chunk of tissue, or this big area of tissue, I can go as high as possible. So I can get like very compact way to classify. One of the most difficult parts to use this tissue classifier is because that you can only enter a certain amount of data. So it's good for very small data with high contrast, for example, in color. And you can see with the real-time tuning that it classifies the areas of my in my region of interest. For example, I enter the class cold glass, the white matter, the gray matter, and the blood vessels, because for me is important. So if I go to the advanced menu, I can go, I can obtain the classes into a mask or in a probability map. For example, if my region of interest, or the most important region for me to analyze is the gray matter, I can get the probability map threshold to tell the computer to exclude those regions that are below that specific threshold that I denotate here. So as you can see, if I increase the threshold here, I can see that the computer is picking a lot of the region of interest, in my case, the gray matter. So also, other option that the computer gives you is create the annotations. So I can let the computer, once you do the classify, please do the annotations for the gray matter and, for example, for the white matter. So I don't need to draw those annotations manually. So as you can see, it's pretty useful, easy to train. However, it has a little of amount of area that you can use to classify. And I can use it only for separating very in high contrast color of my stains. So, but I cannot use it, for example, in this case. This is an LFP, and LFP is a stain for myelin. And as you can see, there are small patches on the white matter that is not homogeneous. It's not a stain that is clearly very homogeneous. So I cannot use the tissue classifier. So I need to go further. The best way to go to this approach is the MiniNet. In the MiniNet, MiniNet is a deep learning approach. In this one, I can use it to differentiate those regions and I can feed the system with more data, because in the deep learning, the more data, the better. However, MiniNet, as their name indicates, has certain limits. So those limits are, so for example, if I wanted to do the analysis of the white matter and include this in the white matter, and telling the computer that this is the white matter, but it's a little bit pale. So I go and train that one. I train the computer to recognize the white matter and the versus the, oh, I forgot this one. So in this case, I can train the computer adding more information. So it's important when you use deep learning that you enter the classes in from the least important to the most important one. Because when you do that, he's going to classify it in that exact same order. So I create those classes here, where it can recognize for example, the blood vessels and separate it from the glass, and give me the information of the gray matter, but also separate it from the white matter. And if you can see, I can enter a lot of data on in this classifier. So I can go here and check here if the class is being separated. If it's not being separated in the way I want it, I can, for example, enter more data, more information. I can run the classifier, and I can start doing the training. So for example, I can say, you are not recognizing, you are not recognizing the gray matter here in this case. So I select the gray matter and start the training. And in here, when I use the deep learning approach, I can obtain two different data that are really important in this aspect. The iteration with in this one, it gives me the information that refers to the number of images patches that passes through the neuronal network during the training. So there are, like, for example, 1,500 patches that are passing right now during the in the neural network. The cross entropy it refers to the difference between the network's classification and the input that I gave and my user definition of the training regions. So the closer it gets to 0 or lower to 0.1 is closer to what you decided is the regions of interest. You can use this one for a specific the type of stains that are very difficult to separate just with manual annotation. And as you can see, while the computer is recognizing, you can see that it's getting a closer approach to what you decide is the best approach to separate those regions. So you can navigate away and see if it's getting closer and closer, and the more it gets closer, it will change that information eventually. So so the more you feed it, the better. The other type of classifier is the dense net. In this case, I use the dense net, for example, I use the dense net to classify it and separate, for example, the different layers. Sorry. Of the cortex. So I can get, sorry. Sorry, my computer is not working right now. Okay, So I use the DenseNet to get annotations, for example, to separate the layers of the cortex using the NUN as a reference. As you can see, the aspect here, I can separate the different layers. I can train it using several annotations, and immediately it will recognize those annotations and create those annotations for me. So I can see and I can verify that those annotations are being created, but the way I wanted to that the computer recognize those layers. So and the good thing about training the DenseNet, the more data you can feed the system, the better. So eventually, if you wanted to analyze 800 slides using the same classifier, you can do it. So you can obtain the different layers of the cortex, and you can replicate those data by synchronization using the serial annotations to get that classification and the same aspect, for example, in this case, to analyze the different plaques present in the different layers of the cortex. Other very useful classifier here is the nuclear segmentation. So in the nuclear segmentation, you can train your computer to obtain a different, not the specific round nuclei, but you can, as you can see, you can train it to recognize the irregular shapes of the nuclei. So for example, for me, it's really important to know how many cells have the cytoplasmic stain for the 88. I can train the computer to recognize those different aspects of the of my specific tissue and in a specific region of interest. So if you can see here, it can start recognizing the cell shape according to the parameters I feed him. So it can recognize pyramidal cell shapes and the negative cells, for example, the ones that are only marked with hematoxylin. And those are really important aspects when I run the parameter and I want to differentiate between the results from neurites and the ATA staining that is localized in my nuclei or the cytoplasm. So, and other very important also classifier is the quality control classifier. So in this one, for example, sometimes the pathologists, they annotate or they can mark, for example, with this in a specific things in your tissue, and it can mess up your analysis. So when you have to deal with this or with imperfections like this, you can train the computer to recognize and separate those from your normal tissue. So if you can see here, you can go to this specific area of your tissue, and you can see that is recognizing what I call abnormal tissue in red and what I call normal in blue. So you can train that, for example, to when there are a lot of debris and your, for example, your immuno is very specific or have a very specific small stain pattern. So you can train to recognize and separate the non-specific staining without interfering with your results. So those are like the brief summary of the Halo AI modules, and I'm going to show you how to combine these modules with the use of other modules in with the other modules in in your analysis. So Halo has different modules, as I mentioned before, and I have the area quantification module. For example, this is the most basic, and I'm going to explain some aspects of these specific modules. One of the important aspects when you do the analysis is the use of the image zoom. So the zoom indicates the resolution that you want to run the analysis. So in this case, my image is scanned 20x, and the vast majority of I would say the 100% of the modules are calibrated to run at 20x. So I have to enter the image zoom as 1. When I select the stain, for example, I need to go here and select the pixels that indicate, sorry, selection. How many stains do I have in my tissue and my region of interest? So I have in this case, the NUN, and I can pick specifically the pixels that I need to recognize. For example, in the DAB, you can hover over the slide bar and you can select the stain here, and the machine automatically averages the amount, the RGB intensity and the optical density, so you can obtain a good color deconvolution. So if you wanted to test if the color deconvolution is good, is enough and sufficient for you, you can select here the real-time tuning, and you can select, for example, the separation in between, and you can play around with those pixels to get the best color deconvolution possible. Also, the machine gives you, when you run the color deconvolution, gives you a pseudo-fluorescent image. As you can see in purple, you can stain that the hematoxylin, and in orange, you can obtain the IPA1. So you can test and check the color deconvolution module here. You can run the area quantification using this pseudo-fluorescent image, or you can do it also with your regular enzymatic reaction, as I show you here. So once you define the color deconvolution here, you can define parameters like the optical density that you wanted to get for your staining and for your analysis. And you can obtain a very good approach of the lower optical density to detect those pixels, medium and high expression of your protein of interest. And in the case of the classifier, you can add the classifier here. So here is when the Halo AI combines with the Halo modules. So you can select, for example, if you are interested in the selection of the gray matter versus the white matter, you can select the classifier you use to design for your region of interest and separate, get the data separated from the one that you want, that you obtain before. So you can add here and you can obtain the data depending on the class specifically. So for example, if I wanted to do the classifier here, and I can do the classification just for the gray matter. And when I run the results, I will get the results just for the gray matter. Other module that is really important for us is the Multiplex IHC. So in the Multiplex IHC, sorry, it takes me some time. In the Multiplex IHC, you can obtain, for example, besides the stain selection, you can detect the nuclear, use the nuclear segmentation. In here, I can combine the AI that I create for the nuclear segmentation here, and I can select what exactly nuclear segmentation I want it to run. In this case, I'm gonna run the nuclear segmentation for ATA staining. For example, if I go to the ATA staining, I can go here and say to use my nuclear segmentation segmentation to obtain information only for the ATA that is expressed only in the nuclei, to get that information separated from the ones that are present in the parenchyma or the neurites in the gray matter. Here you can separate and enter the parameters for to say the computer that you get the nuclear segmentation, also the nuclear for to detect the hematoxylin. And you can also here tell the computer that you only need the information for the certain size of cells or certain size of nuclei, to differentiate, from example, for the small cells that are not important for our study. Also, you can enter the information of the nuclear roundings or the segmentation aggressiveness, which is really important to separate. For example, if the computer sees this as a big cell, you can say, I wanted to divide it in two different cells. So as you can see, it will divide it into cells. And here you can say to the computer that the detection in ATA is very important for you. Here also gives you the information for the threshold of complete, the threshold that is important for your aspect of the analysis in the staining, and the type of analysis that you wanted to obtain. For example, in here, I'm gonna say that those cells are active and positive for ATA, so I can show you that it's recognizing only the cells that has the expression in ATA inside. And you can add here as well the classifier. So if you wanted to run the gray matter, the separation between gray matter and white matter, you can do it here as well. Another important classifier, another important module, sorry, is the object colocalization. So in the object colocalization here, you can obtain the information of these specific cells. I just run it, and you can see the difference between the plaques that are stained in brown and the eyeball cells that are stained in blue. So you can see that in some cases, it will give you the information for the plaques, in some cases just for the microglia, and also it will give you in yellow, for example, the information of the object colocalization when the two meet in a specific area of interest. And the final module that we use in our projects are the microglia activation. So I'm gonna move over to the microglia. And I want to show you the microglia activation. So in the microglia activation, you can enter the information. Sorry, forgot to load it. I'm so sorry. Sometimes, thank you. So in the microglia, you can in the microglia activation, there are the most important aspect is the nuclear contrast, the optical density, and the nuclear size. You can enter those parameters to obtain the best information, the closest information as possible. Also, you can get enter the information to detect the cell body specifically made for the microglia and the processes that you wanted to do. If you wanted to do the optical density closer to avoid a specific aspects of the background here and just get the branching of the microglia, you can enter that parameter here. Also, you can enter the information of how thick the processes can be to say, so the computer can say that this is an activating microglia or an inactivating microglia. In this case, these microglia, for example, here show the processes are very thick. So if the computer will say, make the indication that is activating microglia, and in here, the processes are thinner than my parameter, so it's inactivated. And you can do also for using the classifier to detect the microglia in the white matter or gray matter. So that's it for me. And if you have any questions, I will answer those questions at the end.
Thank you so much. That was really great to see everything kind of integrated together. That's wonderful. Did you want to briefly summarize some of the software updates, Alyssa, and then we can transition into the question and answer session?
Sure, absolutely. That way everybody can take a minute or two to get their questions together. So let me share my screen with you all. Okay, and everybody should be able to see a document really quickly. So I'll just take a couple minutes, not much time at all. But this is a complete list of the new updates for our most recent version of Halo. So recently we've released version 3.3. This new release includes some really nice capabilities, such as a few that you've just seen in Jeanelle's presentation. Additionally, there's an ability to now export your data in FCS format. There is an ICC profile color correction capability and an updated advanced export menu. Related to our Halo AI add-on, Halo users now have the ability to leverage our nuclei segmenter and nuclei phenotyper AI networks in analysis algorithms such as the Multiplex IHC module, the Highplex module, and our RNA scope modules as well. We've also added the ability to now run a test, train, validate workflow on your Halo AI classifiers. And we've also added an additional data output for the nuclei segmenter network. So as always, if you guys would like to speak directly with an application scientist about Halo or Halo AI, or if you have any additional questions at all, please always feel free to reach out to us at our support email, which is support@indicalab.com. So I'll hand it back over to you, Maggie.
Thanks so much. That's great. So I don't see any questions quite yet, but I could start with one of my own questions. Just from some of the more experienced Halo users, can you comment on, it looked really nice, Jeanelle, when you were showing the colocalization specifically for the two different markers. Do you do that with the co-registration, or are you doing multiplexed DAB staining for that?
I use it using the object colocalization module, and in this case, in our case, we use the DAB with the alkaline phosphatase for the microglia, for example, and the DAB for the 6010. And I run that algorithm with the using the object colocalization because with the object colocalization, you can use for irregular shape, like for example, in the 6010, some plaques are very small and other ones are really big. So the object colocalization gives you the exact shape of those plaques and can differentiate perfectly well from the ones present in the microglia cells.
Whether on sequentially cut slides or for the micro, or you're just using for the microglia and the 6010, are those sequentially cut kind of stereology sections? Or
for one of our studies, yes, we use the NUN and to train the algorithm to read the different layers of the cortex, and then we can transpose those annotations in exactly the same position in the tissue using the serial section manager, and we can get the analysis from the every specific layer of the cortex.
Great.
Yeah, just to maybe follow up. So the duplex stains we've chosen that Jeanelle really did amazing work developing was right now is IBA1 and 6010, and then tau and TDP43, and then we have alpha-synuclein and GFAP duplex also, not so much as interesting as the other two sets of duplex stains, but we cut serial sections, adjacent sections from every block, and then they get new in, and then the duplex stains and then unstained slides are saved for future needs.
Great, thank you. So I have a question here. With these classification algorithms, can exact instances be shared so other institutions can test generalizability, or are they specific to the institution who has that specific Halo instance? So for example, do images from multiple studies have to be within the institution who has the license? And if they can be shared, are algorithms retrained / vanilla tuned, or can you treat additional cohorts as holdout set?
So the way we share in some way the algorithms that we develop for our specific needs is using Halo Link. So with Halo Link, we can give to several users, depending on how many users we pay to share with our images. They can run those analyses using, for example, if we upload those images from their specific study, they can run those specific algorithms in those studies. That's the way we do it, but we hold the rights to give the user the ability to annotate, to view, or to have other edition rights.
Okay. Did you want to comment on that further, anyone else? Or maybe Dr. Huber, how your group has done any sharing, or elaborate on that?
Yeah, so we've been, we put together our algorithms. Most of our work is in fluorescence. So I mean, I'll be talking more about that later, but so once you've generated an algorithm, then you could potentially move it from one computer to another, so someone else could use it. There's obviously rights issues involved, but that's something that we can definitely do, and we have moved it certainly from computer to computer to look at different things.
Could you, I don't know the answer to this, and maybe Alyssa does, or maybe Jeanelle does. So the algorithms that we're developing at UW with our license that we pay for, if someone else buys their own license, can we send them our algorithms to run on their slides? Or on their images? Is that something that can be done?
Yes.
Yes. So for example, if we do the parameters and they have a Halo, we can export those specific parameters so they can run those specific algorithms in their studies, but they have to have the Halo to run them.
Right. So just so because it takes me a little longer than other people. So if I could just get this right. So basically there's a couple options, right? So Jeanelle and her team have spent a ton of time developing these algorithms, and they'll be run on hundreds and hundreds of slides from our cohorts. But there's two options. One would be for us to give someone access to our license and our seat and upload their images to our, basically we're using AWS for most of our images, and they could then run samples on on using our license on their sample as kind of a collaboration, or alternatively somebody owns their own license with the appropriate software, and then the algorithms can be just transferred to them. That sounds like the two main options.
Yes.
Thank you for clarifying that. That's very helpful.
And then I did want to add just one thing. The modules that Jeanelle's developing and Dirk are developing, those kinds of modules are going to be a lot easier to transfer than like fluorescence ones, just because the starting material that you have is a lot different. The data is a lot different. So IHC is going to be a lot more transportable between labs.
Yeah, and I just I just wanted to say also, we have been focused on IHC just because of the autofluorescence challenges that you have in brain samples. We haven't tried to tackle that yet. So there's a world of opportunity for IF, I think, with this stuff, and I'm sure Russ can talk a lot about that. With the sort of high throughput that we're trying to do with, you know, hundreds and hundreds of cases of multiple regions, we just decided that rather than trying to develop quenching protocols and that sort of thing, we were just going to run IHC with adjacent slides in duplex stains, maybe triplex at some point. But I think there's a huge opportunity with Halo with IF. We just haven't taken that on yet.
That's great. So I have a couple more questions here. One is, great webinar. Thanks for doing it. Can you get durable with regard to fixation, NUN, antigenicity?
Yes. The most important aspect with human image analysis is having to tackle various aspects of the pre-processing of the tissue. And fixation is a problem when you, for example, run a certain immunos for a long store tissue in paraffin. So the way we do it is to raise that using other products from other companies during the antigen retrieval. And we have great success using one of the companies that is called BioCare Medical, and we use the pressure, the declocker, as they call it, is based on pressure. It keeps steady the pressure, the temperature. It doesn't damage the tissue, and it opens off the antigens even for tissue that is being stored for more than a decade in paraffin, or that was overfixated with formalin. So we haven't had a very great success using those products for antigen retrieval, but that's the most important aspect.
Thanks.
I also wanted to add one thing to that thought. Another thing that you oftentimes will run into is that some of the DAB or the chromogens don't actually last that long. We're using red chromogen for a long time, and you only have a few months in which to get an image of that. So just be aware of that there's a constant breakdown on those things. Other stains are better, but certain ones, and especially the colorimetric ones, can be a little bit they can go away.
Yeah, I think that's an extremely important point. We've had some challenges, and it actually changes as far as I understand, depending on which primary antibodies you're using too. So it's important once you have your stains to have a standard scanning workflow so that you're getting your scans at a timely and consistent way from your slides, because while DAB tends to be pretty stable over long periods of time, as everyone knows, we found the same thing. Some of the alk-phos-based chromogens can be impermanent, or at least not stable. And so that's a really important point. Your images should, right? Images last forever if you don't lose them.
So I think this is a really nice transition question. Can you comment on limitations of Halo that you have encountered in your work?
No machine is perfect. No, it will give you, with Halo, it will give you like 90% of a success on your analysis. For example, I would like to see, for example, in the microglia module, a specific parameter for the nuclei, because when we do the microglia activation, it only counts in processes for the activation or the non-activated cells. And I would like to use also, in combination with the processes, the information obtained for the nuclei, because sometimes the nuclei, for example, in the microglia, when it's activated, it is bigger. So that's one of the limitations I have found, and I know they are working on the more aspects of the neuroscience research to improve their products. I know they are constantly adding and modernizing all their models and classifiers.
That's great. I was just going to comment that one of the things that we have been working to overcome is how to best multiplex. So it's kind of why I started with that question, just to kind of see in your experience how many sequential slides you're able to have success with, and to kind of learn exactly what the limitations are for both bright fields and IF. Seems to be maybe 3 slides sequentially for us, but I don't know if you can comment on that from even an IF perspective, Dr. Huber, or if you wanted to elaborate more on trial and error from your group at UW.
Yeah, so what we've been doing is we've been using the Opal system, which is set up to do up to 6 antibodies and 1 adapy, basically. And as Dirk was mentioning, this is something that you really run into the autofluorescence issue right away. So you have to kind of deal with that from the get-go. There's 2 common methods that people use. One is using some kind of a quencher, and that works. You can use that. The other way is just, since you're doing spectral unmixing anyway, you can use spectral unmixing to eliminate the signal of the autofluorescence, and that's what we've been doing. So we've been using a Vectra Polaris system, and it does that all kind of automatically. And it's a great system. The only thing that I would prefer is if it were a confocal, because then you would be able to get a little bit more high-res data. But despite being not a confocal, being a bright field, it's quite good. It's in terms of spectral deconvolution, it does a great job. We're able to get all 7 colors, and sometimes we'll go with maybe 5 colors just to make it kind of easier, but we have definitely gotten 7 colors at one time. We are doing screens of all of the different astrocyte antibodies that we work with, and then also pathology. So it's been really great. But again, you definitely have to have the right equipment for what you're doing to make the fluorescence stuff work.
Oh, that's great. Thank you. And have you successfully been able to do like the 7 colors on 2 sequential with the co-registration?
We actually haven't done that. We haven't done that.
Okay.
One of the things we were looking at instead of doing that kind of sequential processing was actually to destain the slides and then restain them. And there's another group that's been doing that. And we're going to try and see that, but it's still kind of in development at the moment. It would be great to do sequential slides like you're saying, but I think for the colocalization, some of the things we're doing with colocalization, it's been working better to just use the same slide and just do multiple colors.
Great. Thank you. Very informative.
There's some stuff going on in the chat, but I'm not sure if that's being seen by everyone, Maggie. I don't know if
yeah, I'm back to it now.
I don't know if it's going just to panelists or to the whole group.
Oh, okay. Here we go. Sorry, it was in the other QA. So questions regarding costs of Halo and modules, which I believe Alyssa has addressed, somewhat customizable depending on your group's needs. Next question, what sort of storage, example, GB, TB, are needed per usual case? Can actual computing with groups of multiple cases and images be done without a cloud-based computing environment?
I can speak to that a little bit. The answer is yes. It really comes down to your computer's hardware. So Halo is very flexible with regard to how you run. If you want it in a cloud environment, we can certainly do that for you. But most people do run Halo off of a local instance. And we do have recommended hardware requirements documentation that I can share with you. If you just send us an email to our support@indicalab.com email address, we can have someone send that over to you immediately.
If I could just add, Jeanelle mentioned this, we're scanning basically everything at 20x. Most scanners have a 40x option, but the scanning time is the major limitation from that perspective. It just takes so much longer to get a scan at 40x. And then the file sizes, of course, are the other sort of challenge with 40x. Jeanelle, I think, commented on this, but maybe commented further, and I don't know how it works for IF, but you know, the 20x scans have been sufficient for everything that we've tried, including TDP-43. And so, so that's kind of our workflow. Again, it's kind of a high throughput, hundreds of slides at a time type of a workflow. But if you want to scan everything at 40x, it changes the metrics quite significantly with respect to file size and time.
Yeah, I can elaborate and comment that, yeah, the file sizes can drastically differ depending on, yeah, magnification, your area of interest, bright field versus IF, how many channels. So yeah, it's quite variable.
I do have a little information on that. We were doing a lot of regular bright field scanning with our older Aperio system, and that was the images there for a whole slide. And we do kind of fill up the slide in neuropathology, so just be aware of that. Those were about anywhere between 250 to 700 megabytes. And then when we went to the new, so we just got a GT 450 scanner, and apparently those are actually a lot larger images, and I don't know exactly, but I think it's on the order of maybe 4 times the size. Now, when you go into immunofluorescence, if you're going to multiplex immunofluorescence, you're talking about probably between 20 and 40 gigabytes per slide. So it's a lot of image to move around. I'm happy to say, though, that the Halo has been able to work. It doesn't seem to matter how big our slides are. It does a great job, both in real time and then afterwards in doing the analysis. It's great for that.
Awesome, thanks. So there's another question. Can Halo work with MRX files?
Yep, so I'll speak to that as well. The answer is yes. You just need to also have, so you need to have the MRXS file as well as the accompanying .dat files in the same location, and then you can just upload the image right to Halo, no problems.
Thanks. And then just to wrap up here with a couple last questions. In the chat, there were some questions regarding, does Halo keep track of algorithms? Is there a database available to have a list of the most useful algorithms to share with others along with the copyrights? And Alyssa says, yes, there is. Halo does retain this. So I don't know if you can maybe elaborate on that a little bit.
Yeah, absolutely. So we do have a database. So when you're working with Halo, if you run analysis using any analysis algorithm or a classifier, that information is retained, even if you don't save it, as long as you've run the analysis. So it's nice to, if you forget to run your, save your settings, you can always go to your results and reload your inputs to save that analysis algorithm. And all of that information is contained within the system until you actually go in and delete it.
That's great. Then I have a question or maybe a comment here. What can affect runtimes is what scanner you are using as well, since resolutions can differ with respect to patches. So kind of related to what you're talking about with the different scanner, Russ. So,
Well, I say the question again. What was it?
I think it was more just a comment, just saying that what can affect the runtimes is what scanner you are using as well, since resolutions can differ with respect to patches. So.
Yes, it's definitely true. The only other thing to mention is that, like I said, the performance doesn't seem to be hit by it. The load time can change a bit, but it doesn't hit the performance depending on the size of the image.
Great. Well, I think we're at the hour. I just wanted to thank all of you for your participation today, both those of you attending and in particular, a big thank you to all of the panelists. I really appreciate you taking the time and participating, helping answer some of these questions. And I just wanted to wrap up by announcing to the group that our next session will be September 13th, 11 a.m. Eastern time. And this is going to be session led by Pete Nelson from the University of Kentucky, who will be presenting on quantitative digital pathology methods applied to neuropathology. Instead of kind of focusing more on the algorithms from a more technical approach, it's kind of a more broad overview that will focus on specifically developing and understanding questions in science that can be answered using digital neuropathology. So thanks again, and hope to see you all next session.